The groundbreaking gene editing technology called CRISPR has raced from concept to human trials with breathtaking speed. It was first described by researchers Jennifer Doudna and Emmanuel Charpentier in 2012, and it was so revolutionary that they were awarded the Nobel Prize in Chemistry just 7 years later. CRISPR is a kind of cellular toolkit borrowed from the defense systems of bacteria. It holds the power to add, remove, or alter and repair genetic material. This toolkit lets researchers do gene editing faster, cheaper, more accurately, and with higher efficiency than existing methods. In 2019, Victoria Gray became the first patient to undergo CRISPR treatment for sickle cell anemia. She is thriving and free of the pain and blood transfusions that had ruled her life. CRISPR is even being deployed to offset some of the effects of climate change. What used to require a decade or more to breed a plant to make it more resistant to a given threat, CRISPR can do in a year or two. For example, researchers have altered the genes of corn to grow at shorter heights to better withstand storms. And they have genetically engineered rice to resist 95 different strains of bacteria that cause blight. In humans, more than 50 clinical trials of CRISPR are underway. A dozen of them have advanced to phase 2. They target leukemia, lymphoma, HIV, myeloma, sickle cell anemia, bladder cancer, and more. In China, CRISPR is being tested against lung cancer. And in a first-of-its-kind case last year, a patient with congenital blindness received an injection directly into the eye, using a virus to deliver CRISPR to the retina in the hopes that the enzyme will repair a defective gene and restore vision. Plus, new CRISPR-based diagnostics are being tested for pneumonia, tuberculosis, whooping cough, and COVID-19. CRISPR is an acronym that stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's actually a technique borrowed from an ancient defense system bacteria used to defend against viruses. The CRISPR complex consists of a strand of RNA bound to an enzyme called Cas9, which essentially unzips the double helix structure of the invading virus's DNA and chops it up. The power of CRISPR lies in the specificity that comes from lab-synthesized RNA, called the guide RNA, that will determine exactly where the gene editing will take place. Once the CRISPR toolkit reaches the cell's nucleus, it bumps along the genome, attaching every time it comes across a short sequence known as a PAM. At those PAM entry points, the CRISPR grabs onto the cell's DNA so the Cas9 enzyme can unzip it just a little bit. Then the guide DNA slips inside this unzipped double helix to look for the target sequence of DNA it seeks to repair, rewrite, or delete. If the guide RNA fails to find the right sequence, CRISPR moves on to the next PAM entry point and the next until it finds a precise match. Then the Cas9 enzyme cuts the DNA right where researchers were targeting. Then the cell's natural DNA repair mechanisms swing into action. Cells usually repair a break in their DNA by gluing the loose ends back together. It's a sloppy process that often results in mistakes, but mistakes can be beneficial if they disable or inactivate a disease-causing gene. CRISPR also lets researchers insert a custom-made DNA sequence into the newly split gene. It serves as a template as the cell repairs and reforms its double helix. That's essentially a new gene that could code for a vital, missing, or defective protein. Cancer is another CRISPR target, using gene editing to engineer CAR-T and natural killer cells that boost our immune system to make it better at destroying malignancies. Other diseases that may become CRISPR targets may include cystic fibrosis, muscular dystrophy, Huntington's disease, atherosclerosis, and progeria. The possibilities are endless. This is why CRISPR technology is so powerful and so very promising. But like any powerful technology, CRISPR's potential can be put to fraught uses. We must ensure that humanity's ethics are safeguarded as we explore this potent science. The conversation bridging science and faith has begun.